So, the genetic code. The first one was deciphered by Marshall Nirenberg in 1961. He made artificial mRNA containing only uracil, so U. The mRNA was translated into phenylalanine amino acid subunits. So the genetic code. There are 64 possible mRNA codes. Should be a lowercase m. So you'll notice that it's not really a whatchamacallit. It's not really a Punnett square, but it looks like this is something that if I gave this to you, I'm not going to expect you to remember this, obviously, but you need to know how to use it correctly to interpret it. So we have a series of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Oops, I count those a couple twice. So notice how there are a couple ones that are kind of redundant. Like you have multiples of the same one. I don't know if you realize that. But you have a start codon, methionine AUG, and you have 3 stop codons. That means that these 3 codons can signal a protein to be not made anymore. And you notice that each, like threonine has 4 different possible combinations. Arginine has, actually, look, if you notice, it has 6 possible, same as leucine, 6 possible combinations. So the reason why I'm showing you this is because there's a chance you're going to have to be able to code, or at least, decipher what protein is being made, what codons are being amino acids are going to be made. So kind of keep this in mind. We'll work on this in class, most likely. So like I said, why do we have multiple codons for the same amino acid? Let's think about it. Why would I have multiple ones for the same thing? Last chapter and other chapters we talked about mutations. Look how for serine, for example, right here, if you just change one of these at the end, you can still end up with potentially the same amino acid, which is good. There's some redundancy, so in case there's one small genetic mutation, you still end up with the same amino acid. So what is translation? Translation is defined as a process that takes the information passed from DNA as messenger RNA and turns it into a series of amino acids bound together with peptide bonds. Okay, notice how I'm starring and circling all the bonds, so you're going to have to be aware of those. What does RNA do in translation? It takes place in the cytoplasm, and tRNA and ribosomes help in the synthesis of proteins. Each tRNA is folding into a compact shape and has an anticodon. And it's a 3-nucleotide sequence on a tRNA that is complementary to an mRNA codon. That's a lot of words, if you will. The amino acid that a tRNA molecule carries corresponds to a particular mRNA codon. There are 3 phases of translation. The first one is initiation. The ribosome assembles around the target mRNA, and the first tRNA is attached at the start codon. Remember, the start codon is AUG, and that's short for methionine. So make sure you know that. Elongation. The tRNA transfers an amino acid to the tRNA corresponding to the next codon. The ribosome then moves/translocates to the next mRNA codon to continue the process, creating an amino acid chain. Termination. So this kind of makes sense. When a stop codon is reached, the ribosome releases the polypeptide and you're good to go. So what are the stop codons? We'll talk about that in class. The polypeptide forms into its native shape and starts functioning as a functional protein in the cell. Okay, intervening, I think we're pretty close to the end for this video, so it's going to be 2 videos, and I guess because it's over 13 minutes and I'm talking right now. So, introns versus exons. Just a couple of some terminology we should be aware of. Introns are long segments that have no coding information. Exons are portions of a gene that are translated into protein. So the way that I remember it is exons are expressed. After a eukaryotic gene is transcribed, spliceosomes cut out introns. So they're spliced out, they're cut out. The exons are then spliced, stitched together by the spliceosome to form a smaller mRNA molecule. So why have these exons and introns and all that? So the whole point is to be flexible, evolutionary flexibility. So cells can shuffle their exons between genes and make new genes. One gene can code for several proteins, and they'll have different functions. This allows for a shorter chromosome so we can be more efficient, if you think about it that way. All right, so this is the end for part 1. Hope you all somewhat enjoyed this lecture. If you have any questions, let me know, and I'll see you all later. Take care.